All right, so there's just a couple more slides in this lecture. And then we'll talk about the quiz, which we haven't talked about yet, and then that diffusion worksheet, which I didn't grade yet. If you turned it in on Wednesday, I'm sorry. I'll try to look at them today, right after class. But a lot of people turned them in, so I did go ahead and grade those that turned them in. And I kind of mentioned, well, we'll get into that in a minute. I'll jump ahead. And then we'll do the Kahoot review quiz, and I can answer any questions you guys have regarding the exam or the material. So first, let's do this. All right, so I mentioned there were 3 checkpoints you want to be aware of. So this cell cycle hopefully looks kind of familiar by now. So we have mostly, remember, a cell mostly stays in interphase. There's a couple of checkpoints during 2 stages of interphase, the G1 and G2. One really important aspect that's done in both of those checkpoints is checking for any DNA damage or errors. If there's any problems with the DNA, it's checked for at the G1 checkpoint as well as the G2 before that cell actually divides into 2 cells. In addition, at the G1 checkpoint, the cell is checking to make sure it's big enough basically, and it has enough nutrients to kind of continue to grow. DNA is then, well, so this resting phase here, if any of these are a problem, so say there aren't enough nutrients, the cell can go into this resting phase where it just continues to grow and take on more nutrients. So that's the G0 that's shown here, the resting state. If there's enough nutrients, it's big enough, there's no DNA damage that can't be repaired, it'll continue on into the S phase, the DNA synthesis phase, which is where the DNA is copied or replicated. And then at the end of G2, cell size will be checked for again. So is the cell big enough to actually divide into 2 cells? And also, did the DNA replication go okay? Are there any DNA errors? You don't want to divide a cell, basically copy a cell that has DNA errors, right? We don't want to continue that erroneous hereditary information. Okay, I won't talk about what happens if there are DNA errors, but there's a few different ways that a cell can fix those. We'll say everything's good at the G2 checkpoint, so that cell then starts the M phase or mitotic phase. There's a checkpoint in there as well, sort of, I guess right in the middle, in metaphase. So if you remember metaphase, that is where the chromosomes are lining up right along the middle of the cell. And then these spindles are attaching to either side of the chromosome, and then they'll be pulled apart to either side of the cell. So what the metaphase checkpoint is checking for is whether the chromosomes are appropriately attached to that spindle apparatus that will pull them apart. If they're not correctly attached, each cell might not have the right amount of DNA. So 2 chromatids might end up going to one of the cells, whereas the other eventual cell will get 0. So having those spindles attached correctly ensures that those chromosomes, which each have 2 copies of the DNA, will be split in half perfectly. So those are the 3 checkpoints you want to be familiar with. I'm not going to go into a lot more detail, or really any more detail, on the basics of those checkpoints. Know their names and then what they're checking for. Those are the main points you want to take from this slide. A really important aspect is checking for DNA damage at G1 and G2. Okay, so those are the checkpoints. I think I ran through them really fast at the end of class on Monday. And just very briefly, I think most people have some understanding of what cancer is, but cancer is basically when the cell cycle, the cell checkpoints are not working correctly, and you have essentially uninhibited cell division. So cells just keep dividing over and over, and that's what actually creates the disease that we call cancer. So yeah, these cell cycle checkpoints, there's some error where the cell is not it's continuing to go through mitosis even though it shouldn't. At the checkpoints, like I said, there's a couple different things that can happen with the DNA. It can either be repaired if it's not that bad. So say at the G1 checkpoint, if there's some kind of DNA damage or error, the cell can potentially repair it. Or the cell, if it's kind of beyond help, the cell can basically die. It's called apoptosis. And it just doesn't continue on. So that's ideally what happens in the situation of cancer. Neither of these occur. So the damage isn't repaired, but the cell also doesn't die. And these cell cycle checkpoints are essentially controlled by what is called a tumor suppressor gene, the p53 gene. Genes have all these kind of complicated numerical and alphabetical names. So the p53 gene is the tumor suppressor gene. If an individual has a faulty p53 gene where it doesn't actually suppress tumor growth, then they're more likely to potentially get cancer. So you can screen people genetically to look and see if their p53 gene is active and doing what it should be doing. And I have a little video here from, I don't know if have I shown you guys the Amoeba Sisters yet? I think I have. Just an interesting video kind of about how this cell cycle kind of going out of whack can cause different types of cancer. So we'll watch this real quick. I think it's only like 5 minutes. All right, stop share.
When school isn't a place you have to be, why not go to school where remote learning can be as remote as you want?
Have you ever been sitting in class and thought to yourself, I wonder what my skin cells are doing right now at this very moment? This kind of pondering may be unique to me, maybe, but wouldn't we at some point wonder what our cells are doing right now? Because if you remember, as part of the cell theory, you are made of cells. All living things are made of one or more cells. Many multicellular organisms like you have cells that work together, working together as part of a body tissue working together as part of an organ, organs working together as part of an organ system. Your cells are specialized to work in these different levels of organization. You have skin cells, stomach cells, muscle cells, just to name a few, and their functions need to be regulated. These cells actually are regulated as part of something called the cell cycle. And that is going to relate to my question of, I wonder what my cells are doing right now. Cells themselves can grow in size, but let's put it in perspective now. A multicellular organism isn't growing because each individual cell is getting bigger. A multicellular organism itself grows by making more cells, by the cells making more cells by dividing. That's cell reproduction. One reason that you're bigger than you were when you were 5, unless you are 5, is because your cells have divided to make more cells. Mitosis and the cytokinesis that follows that splits the cytoplasm allows you to make new body cells. But you don't want that cell division happening all the time. Why? It is likely that you've heard the term cancer before. We have had family members that have battled cancer before. It is definitely a relevant topic for all of us. Cancer is in part due to cells that divide too frequently. The cells are not regulated. They are uncontrolled. Cancer cells can have other problems too. They might not be able to communicate with other healthy cells. They may not be able to carry out normal cell functions. They may not securely anchor themselves like other cells do, which can make them more likely to travel somewhere else. Some cancer cells have the ability to secrete their own growth hormone that makes blood vessels divert over to those cancer cells and supply the cancer cell with nutrients, which can take nutrients away from healthy cells. Why do cancer cells become this way? Well, there's a lot of research in this area. With some cancers, there may be genetic links, making some cells more susceptible to having problems. These genetic factors might run in families. Exposure to toxins, radiation, excessive exposure to UV light, all of these can be risk factors for some cells to become cancerous. The uncontrolled growth that cancer cells have gives rise to more cells like them, which can develop into a tumor. Some tumors stay put, but some do not. Now, the good news is that scientists continue to develop better treatments, which include destroying the cancer cells with radiation or medication, such as chemotherapy, which will target cells that divide frequently. Maybe someday you will be part of helping to meet the challenge of trying to eliminate cancer, because the fact remains that these cells are not participating in the cell cycle like they should. So what is the cell cycle? The cell cycle is often represented in a pie chart like this. Cells are either in one of two different phases, a phase called interphase, where the cells themselves are growing, replicating their DNA, doing their cell functions, or they are in M phase, which includes mitosis and the actual splitting of the cytoplasm, cytokinesis. So this M phase is where cells actually divide to make more cells, but cells spend most of their time in interphase. So most of the time, they're not dividing. Now, depending on what kind of cell, it might do mitosis more or less often. For example, your hair follicle cells do mitosis frequently, which is why your hair can grow at the rate that it does. It's also why many cancer drugs may also target hair follicle cells, because many cancer drugs go after cells that do cell division frequently. It's a big deal for cells to hit this M phase. If a cell has an error, a harmful mutation, for example, you do not want it to divide, because then it will create another cell that has the same issue. That's where checkpoints come in handy. Along the cell cycle, there are checkpoints to check that the cell is growing well and replicating its DNA correctly and doing everything it's supposed to do correctly before it divides. To better understand those checkpoints, let's further divide the cell cycle pie chart. We have G1, S, G2. All three of those are part of interphase. Then we have M phase, where mitosis will happen. During G1, the cell individually itself grows. Then it replicates its DNA in S phase. You can remember that because the S is for synthesis, which means to make something, and it's making DNA. Then G2, the cell grows some more in preparation for mitosis. So let's take a look at checkpoints. We've got one here in G1. This checkpoint checks, is the cell growing well enough? Is its DNA damaged? Because if it is, you definitely don't want it to move on to S phase, where it would replicate DNA. Does the cell have the resources it needs if it were to keep moving on? This checkpoint in G2 checks if the DNA was replicated correctly back in S phase. Is it growing well enough? Does it have the resources it needs to continue? Okay, then moving on. This next checkpoint in M phase is my favorite checkpoint. It checks in the stage metaphase to make sure the chromosomes, which are made of DNA, are lined up in the middle correctly, that they're all attached to the spindle correctly, because if they're not, the chromosomes will not be separated correctly. So now you may have two big questions. First, what happens if the cell doesn't meet the requirements of the checkpoint? And second, what is doing the regulating of this cycle anyway? To address the first question, if the reason the cell can't go past the checkpoint is a reason that can be fixed, the cell may kind of pause here until it can fix that issue. But if it can't be fixed, then the cell does something called apoptosis, which basically means the cell self-destructs. This ensures that a cell that is damaged beyond repair will not go on to divide. So what is doing the regulating anyway? We've mentioned before that proteins are a big deal. Genes in your body can code for proteins that do an assortment of functions. And there are many proteins involved with regulating the cell cycle. Some of them are positive regulators because they allow moving forward in the cycle, and some are negative regulators that might make things stop. The proteins themselves can be sensitive to cues inside and outside of the cell. So, for example, two proteins that are involved in positive regulation are cyclin and CDK. CDK is specifically an enzyme protein, a fancy kind called a kinase, which is worth a Google. CDK can have different forms of cyclin protein bound to it. Different types of cyclin rise and fall throughout the cell cycle. And the rising and falling is based on a variety of signals to determine when the cell should move on to the next cell cycle phase. Typically, each cell cycle phase, G1, S, G2M, will tend to have a different cyclin binding with the CDK. The rise and fall of cyclin types and the role CDK has when it's active is a fascinating subject to explore. Remember that vocabulary word we said, apoptosis? Proteins that are negative regulators, for example, a protein called P53, can be involved in initiating apoptosis. Again, we encourage you to explore beyond the video. One last thing to mention. There are some cells that don't go through the phases we mentioned because they're actually in G0. That's a 0, by the way, and not a no, because if it was an O, then it's a go, and G0 is kind of the opposite of that. G0 is a resting phase. Now, cells here are still performing cell functions, but they're not preparing to divide. Some cells go here temporarily, maybe if there's not enough resources around, for example. But some, like many types of neurons in your brain and spinal cord, may stay here permanently. If they stay here permanently, they'll never get to end phase, so they will not divide. This can be one reason why a major injury to the brain or spinal cord can have challenges with healing, as many of those cells may not be able to replicate, a topic that definitely continues to be researched. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, hopefully that was kind of a good review actually of the cell cycle. I forgot they went into so much detail about the cell cycle. Let me get this sharing going again. Okay. So that was actually kind of good timing. I'm glad I didn't get to that until the day before the exam. I want to mention there's a lot of good stuff on YouTube. I think the Amoeba Sisters, if you guys are confused about a topic, so they have a mitosis video, they have a video about many, many different topics that we're talking about in here. If you guys just need a review, or I was confusing when I explained it or something, I didn't explain it well, go check them out. I think they have some really great explanations, and kind of at the level, more or less, it's going to vary a little bit, but the level that I'm kind of trying to get across to you. So I think they're a really good resource for actually studying for the exam. Obviously, you can't just watch them and not take the material in. So if you take notes during it, I think that can be really helpful. It's just another kind of study tool. All right. So yes, that kind of wraps up the cell cycle. Oh, this is just kind of an interesting disease where the cell cycle also doesn't check itself. I don't know if you guys, have you guys seen The Curious Case of Benjamin Buttons? It's like an older movie now, but Brad Pitt was in it. And he had, in that movie, he had this Hutchinson-Gilford progeria syndrome. And this is basically really premature aging in children. So I guess maybe he was the opposite, didn't he? He got younger. I don't know. I can't remember. He was born old and got young, but it kind of reminds me of that. So this is where essentially children age really quickly. So by 12 years old, their cells have undergone essentially unchecked cell cycling, which is what part of what causes aging in individuals. When your cells kind of divide and divide over and over again, after a while they get kind of tired of dividing, and so it leads to many different symptoms of aging. But this really, really rare syndrome called progeria syndrome is essentially due to one gene that keeps a lot of the cell cycles or the cell checkpoints in your body from behaving correctly, and the cells just continue to divide a lot quicker than they should, so it leads to really premature aging. So anyway, that's just kind of an applied aspect of what happens when the cell cycle, beyond cancer, what happens when the cell cycle is kind of thrown out of whack. So it's a really important aspect to how most of us function. You don't have to know this for the exam. I'm not going to ask you about that. It was just an interesting kind of side note. Okay. And then at the end, I just have kind of make sure you can do these things before the exam. Just a sort of a mini study guide, although the study guide is on Canvas too, probably includes these bullet points. So those are at the end of each of the lectures, I believe. So you should have access to those on Canvas. Okay, any questions about that material? Okay, so that's the end of the material for the exam on Monday. What are we doing now? Let's go over the quiz. Let's do the quiz that was due, was it last Friday? So this will be kind of a nice review. This is obviously just the first half of the material for this unit. Okay, so you guys on Zoom should be able to see the quiz. All right, so the first question, I didn't actually look at the, I didn't look at the item by item analysis to see which ones a lot of you guys missed on this, but I think the grades were overall really well. They were really good. So you guys did well on the quiz. So which of the following is not a tenet of the modern cell theory? You want to know those 3 tenets of the modern cell theory. It's only 3, and they're pretty simple and straightforward. Cells are not necessarily independent entities. They work with each other. We are multicellular, so that is the one that is not a tenet of the modern cell theory. The 3 other ones are. And this quiz should still be available to you, so you can use it to study. Which of the following organelles contains DNA, a double membrane, and converts light energy to chemical energy? So that's going to be the chloroplast. This is one of the organelles you needed to know. I don't think we didn't talk about it in the Cell City activity we did specifically because they're only in plants. So only plants are taking sunlight and converting it to chemical energy. We can't do that. We can't absorb sunlight and make energy out of it, make chemical energy out of it. So that is a chloroplast. That is the function of a chloroplast. They have their own DNA just like mitochondria do. They also have a double membrane. But the main giveaway there is that it converts light to chemical energy. Stop me if you have any questions about any of these, obviously. Okay. As I said, I'm definitely going to ask you about solutes moving and water moving across membranes in response to other concentrations. So this is a pretty big topic. It'll come up over and over again. So for this one, if a solute, so a solute, you can think salt, is said to be moving down its concentration gradient, it's moving from an area of high solute concentration to low. So we're just talking about solute here. We're not even thinking about water. So down a concentration gradient is going to be from high to low. Which of the following organelles is responsible for making proteins? That's very straightforward. So ribosomes. Every time you see the word ribosome, think protein. Which of the following is found only in eukaryotic cells and not in prokaryotic cells? A nucleus. So make sure you look back at that slide that has prokaryotic cell versus eukaryotic cell. That was one of the first lectures in this unit. So all of these are in, well, nucleoid is only in prokaryotic, but I wasn't asking that. So prokaryotic and eukaryotic cells have ribosomes and some kind of membrane around them. Prokaryotic cells have a nucleoid, whereas eukaryotic cells have a nucleus. Okay, and then this one is about the surface area to volume ratio, right? So which cell has a higher efficiency? Cell A, side length of 2 millimeters, cell B, a length of 6 millimeters, and from that surface area to volume worksheet you guys did, hopefully you remember the bigger a cell gets, the less efficient it gets, basically. So the larger the side length is, the harder it is to get enough nutrients into that cell to actually support it. So smaller cells are going to be more efficient. That's cell A. Tay-Sachs disease was something I talked about specifically when I went over lysosomes. Sorry, to split across a couple pages. And specifically, it's a disease that hinders lipid digestion. So lipids will build up in a person's brain that has Tay-Sachs disease because their lysosomes are not functioning correctly. Okay, phospholipids, amphipathic. You definitely want to know what amphipathic means. It means they have a hydrophobic and a hydrophilic region. and know what hydrophobic means and hydrophilic. So hydrophobic means it's not going to mix with water or other polar molecules. Hydrophilic is the opposite. It will mix with water and polar molecules. Okay, so phospholipids, those are a big part of the cell membrane. Then the endosymbiont theory. So this is that video I had you watch again. Yeah, the Amoeba sisters. I have had you watch them before. So they did a video about the endosymbiont theory. And it basically says that mitochondria, as well as chloroplasts, I think, but we'll focus on the animal side. Mitochondria evolved or descended from prokaryotic cells that were once free living. So the idea here is that one cell basically ate what was to become a mitochondria, and that mitochondria, rather than getting digested, just started producing energy for that cell. That's why mitochondria and chloroplasts have their own DNA. They were once free living prokaryotic cells. All right. And then finally, oh yeah, transmembrane transportation. So we talked about stuff moving across membranes a ton, right? The one that requires energy is active transport. The other three types listed here are passive transport. You want to know the differences between those, but active transport is the only one that requires ATP or energy. Any questions about those? I think you guys, like I said, did pretty well on that quiz. So use that to study. You've probably seen that I think I mentioned this, I like to sometimes use some of the questions from the quiz on the exam. So it's a great way to study. All right, then the last thing we're going over. Here we go. That diffusion worksheet. Like I said, if you turned it in on Monday, I think I graded it already. If you didn't turn it in until Wednesday, I will grade it, I promise. But I have not yet. So I want to make sure you know the correct answers here. This is just a picture of my writing, so, oh, it's pretty clear. Okay. So this was diffusion and osmosis. So remember, osmosis is just the movement of water. Make sure we have time for the quiz. Okay. We looked at water as well as solutes moving across the membrane. I didn't say what this was, it's just a molecule. And diffusion is where molecules are going to move from high concentration to low. So it always go from high to low. So there's a lot of molecules inside this cell here. They're going to move out, and then the opposite will happen for B. So if it's just diffusion, the molecules are always moving. The solute molecules are moving from high to low concentrations. They want to get away from each other. Same here, we're talking about sugar moving. So same idea, I just gave you percentages. So sugar is going to move towards where there's less sugar, basically. It's going to spread out. And then with this, it'll be moving. There just will be no net movement. So it'll move in and out equally. Okay. Cell membrane is a phospholipid bilayer. Phospholipid bilayer. You definitely want to remember that term and know what it means. The cell membrane is semi-permeable, or selectively permeable is another term for that. And that means only certain molecules can pass through it. I think most people got that. And here we go. So diffusion is where particles move from a region of high to low concentration, high to low. Osmosis is a little bit different. So we'll talk about that in just a second. Does the cell use energy when molecules diffuse in or out of the cell down the concentration gradient? Some people said in or out as an answer, and I guess this is worded a little bit funny, but I was just asking, is energy used during diffusion? No, no energy is ever used during diffusion. It's a passive form of transport. Okay, so I think people were getting a little confused with movement from what I saw on this worksheet, thinking about water moving. So water is going to move towards the higher solute concentration. Here, I was just talking about the solute itself moving. So definitely make sure you separate those two concepts in your mind. Water will move towards where there's more solute. So into or out of a cell, for example, like we talked about. Solutes, if we're just talking about diffusion, they're just moving from high to low concentration. So it's kind of backwards. Definitely keep that straight. Okay, any questions about that? Nothing, okay. So I'll get those graded and any comments back to you guys. I totally forgot to do that yesterday, sorry. All right, what am I doing? We're doing the Kahoot quiz. You guys ready for Kahoot? So that's the last thing we'll do today. Should have time, hopefully, to finish it and get it up and going. And I have posted the link on Canvas. So if you go into the modules section of the Canvas course, there, I put it in a couple places. It's under the links module, and it's also under the Unit 2 module. (1101) exam 2. Okay, so for you guys on Zoom, I'm going to hold you up to the screen here. There we go. And I will say, as you guys are signing in here, using the Kahoot quiz, using that quiz we just went over, the study guide, which obviously doesn't have answers on it, but it'll at least guide you to sort of what to focus on. Those are kind of the best ways. In addition, well, I guess you can quiz yourself using this, but quizzing yourself on that is a really good way to study. So actually actively engaging in that way rather than just reading through material. It's easy to read through material and for it not to sink in. For some people that works. If it didn't work for you for the first exam, definitely try something different. I always suggest somehow quizzing yourself through Quizlets. There's some Quizlets I think that are already made online probably for this material, or you can make your own. Or note cards, kind of a low-tech Quizlet. All right, we're gonna go ahead and get started in the interest of time. Here we go. What is the primary purpose of a ribosome? I hope everyone gets this. Oh, and I should say, same setup on the exam as last time. 35 multiple choice, 5 fill in the blank. We'll take it in person on Monday over in the bio labs. Oh, one person missed it. That's okay. So a ribosome is where proteins are made. Ribosome equals protein, basically. Okay. Next. Which of the following provides physical structure to a cell? All right, the cytoskeleton. Good job. So the cytoskeleton, just like it sounds like our skeleton gives us structure, the cytoskeleton provides physical structure and it also helps move stuff around the cell. Which organelle will be abundant in flight muscles of birds in order to provide a lot of energy? So a slightly applied question. Check out what's important there. Nice. Yep, mitochondria. Mitochondria are the energy centers of cells. If something needs a lot of energy, it has a lot of mitochondria. Of some area, I should say. Their cells will have a lot of mitochondria. Right. What type of protein assists in the movement of water molecules across the plasma membrane? The aquaporin. So the aquaporin, as it kind of sounds like a water pore, allows water to come through the membrane. The phospholipid bilayer does not want to let polar molecules through, which water is. So water doesn't really make its way through the membrane very effectively. It needs these little aquaporins to help it move through. The other big answer was cholesterol. Cholesterol is important. I don't know if it comes up in another question, so I'm not going to talk about it, but you definitely want to know the function of cholesterol in a plasma membrane. What does it do? Why is it there? The plasma membrane is made primarily of nice phospholipids. Yes. So phospholipids are the main component of a membrane. So when you think of a membrane, think phospholipids. There are a lot of proteins. There are proteins in there, but they make up a relatively small proportion of the overall molecules. All right, so phospholipids. What else? Okay, here we go with cholesterol. Just barely, nice. So membrane B. Cholesterol, what it does is help to solidify membranes. With cholesterol molecules stuck in between the phospholipids, that helps to solidify it, which decreases fluidity. So when I say fluidity, I just mean the movement of those phospholipids around each other. With more cholesterol, it decreases fluidity, making that membrane a bit more solid. All right, number 7. Which of the following is a function of membrane proteins? Okay, cell-to-cell communication. I didn't spend a ton of time on this. There was one slide that basically had a list of the different functions of proteins in the plasma membrane. So cell-to-cell communication was one of those that are listed. The other 3 are not functions of the membrane protein specifically. So make sure you look at that slide and you know those functions. The one we focused on was obviously moving stuff from one side of the membrane to the other. Will a large nonpolar molecule be able to cross the plasma membrane without the help of a transport protein? Yes. So large probably threw you off. If you see nonpolar, a nonpolar molecule is going to move across that membrane just fine because it's basically a nonpolar membrane. So the interior part of that phospholipid bilayer is nonpolar or hydrophobic. Anything that's like that, nonpolar or hydrophobic, can go through just fine no matter how big it is. It gets a little more complicated when something is polar and wants to go across. So definitely look into that. Something that's small and polar can kind of make its way across like water to a certain extent. Anything large and polar, not without a transport protein. And anything that has a charge cannot make it across without a transport protein. All right. How much time we have? Okay. In simple diffusion, molecules move where? Nice. Yes, down or with their concentration gradient. I think I've primarily used up and down in this class, but with and against is another way to say it. So I put both of those there for you, just in case. So simple diffusion, molecules are moving down their concentration gradient, like I just mentioned before. That means they're moving from an area of high to low concentration. All right, Ty, still in the lead. Which process below is ATP needed for? Sodium potassium pump, that's a tough one. So these three are passive forms of movement: facilitated diffusion, simple diffusion, and osmosis. Energy is not needed for any of those. So even if you don't know what the heck the sodium potassium pump is, which you should know what the sodium potassium pump is, hopefully by process of elimination you can figure that out. The sodium potassium pump is moving things against their concentration gradient. Don't know if I have that as another question in here. You want to know how many molecules of sodium and potassium are moving in which direction for the sodium potassium pump, definitely. And know that it needs energy to happen. Number 11. Okay, so we have concentrations. Solution A, 0.5 molar, solution B, 0.8 molar. Solution A is what? Solution B. And those brackets just mean concentration of sodium. Nice, okay. In the right direction, so hypotonic. Hypertonic, hypotonic, isotonic. You definitely want to know those different definitions. Hypotonic means that, so I asked solution A is what to solution B. So solution A has less sodium in it than solution B does. That makes it hypotonic. It has a lower concentration compared to solution B. If I flipped it and said solution B is what to solution A, it would be hypertonic. So it's always in relation. One's going to be hypertonic, one's going to be hypotonic, unless they're the same, which is isotonic.
Okay, so when it's hypotonic, it's less solution.
It's less solute. Yeah, which would be the sodium. Yeah. Yep, low, hypo, low. You held on for a long time. That was good. Oh, here we go. Sodium potassium pump. How many sodium ions go out and how many potassium ions come in? Okay, nice. That I know is really specific, but it's so important to know that 3 sodium ions are going out, 2 potassium ions are coming in. That's gonna, I don't think this is a question I have, but that's gonna make the inside of the cell negatively charged because you have 3 positive ions going out and 2 positive coming in, which is gonna slowly lower the charge of the interior of that cell. That's what creates the voltage in our cells and allows our muscles to contract, for example. Okay, so definitely know those numbers, 3 and 2. 3 sodium out for every 2 potassium in. All right, a few more. What stage of the cell cycle do cells spend most of their time in? Nice, awesome guys. Interphase. So if you think about that pie chart, like we just looked at in that video, most of it is interphase. Mitosis is this little tiny chunk of the pie, right? So those are cytokinesis, prophase, mitosis, those are all in phase, that tiny phase. Interphase is where cells spend the vast majority of their time. All right. 14. How many chromatids are present at the end of G2 of interphase? So like per chromosome, I should say. 2. Yep. So G2 is at the very end of interphase. We've already replicated the DNA, which means there's 2 copies of the DNA. That means there's 2 chromatids. So the chromatids are what come together to make the kind of typical chromosome shape that you think of, that X shape. So 2 chromatids are present, 2 copies of DNA essentially. There's 2 copies of DNA at the end of G2 after that S phase has already replicated the DNA. All right. Last one. Which stage of mitosis is shown in the image below? Anaphase, yep. And the next one, telophase, is close to telophase, so there's no pinching end of the cytoplasm yet, and it still looks like there's still some chromosomes that are moving towards the edge of the cell. So those chromosomes are now chromatids. They're just individual chromatids. They're moving to either side of the cell in anaphase. So definitely be able to recognize those different stages based on cell or microscope slides. Yeah. PMAT? Yeah. Well, PPMAT, yeah. Because there's prophase and prometaphase, but yeah. PPMAT. All right. I once again forgot to bring treats. Sorry. Lincoln number 3. Grace number 2. Number 1. All right, good job, Chloe. Nice job from Zoom. Ty and Allison, close runners up. All right, good job, guys. That is linked on Canvas, and email me with any questions you have. Otherwise, I'll see you on Monday in the bio labs.